let's focus on what the brain does as a whole. The brain, and then towards the end of this presentation, we're going to talk in more detail about the functions of the individual cranial nerves that you learned during lab. So when we think of the brain, there are a lot of higher brain functions. When we think of higher brain functions, typically we refer to brain functions that are associated with the cerebrum, not necessarily the diencephalon or the brainstem or the cerebellum. These are typically going to be cerebral brain functions. And the cerebral cortex is going to interact with other brain functions. As we have learned more about the brain, as neuroscience has advanced, we've slowly been learning and discovering that any one specific function located to one specific part of the brain. In a sense, there's kind of multiple structures that contribute to the same function. With that as a caveat, when we're going through this presentation, we'll talk about which structures of the brain will primarily focus on a given function, but keep in the back of your mind, just as a caveat, that other parts of the brain will still contribute to that function. So we don't have very clear anatomical boundaries to these parts of the brain, or these functions of the brain. Let's first talk about the primary sensory cortex. This is part of the cerebrum that is primarily associated with sensation. And when we say sensation, we're going to think of general senses. So touch, or temperature, or pressure, or light pressure. These are the kinds of things that are going to be processed in the primary sensory cortex. As we look at the primary visual cortex, the association areas, those association areas are regions of the cerebrum next to the primary sensory cortex, and in processing the information. But they aren't the final arbiter. So we can find that there's the primary region of the cortex that handles that information, and then there's other regions of the cortex that help to handle that information. When we look at the primary visual cortex, this is a part of the brain that's overwhelmingly associated with vision. This is the part of the brain where we take those signals from cranial nerve number 2, the optic nerve, and convert that into an actual image we can see. There are other parts of the brain where we have multiple senses, multiple inputs coming in. We call those multimodal association areas. These multimodal association areas help us to integrate multiple modalities, multiple kinds of stimulation: heat, temperature, pressure, sight, sound, equilibrium. We can take all this different information and integrate it together at our multimodal areas. When we think of special senses, I want you to think of sensations associated with the sensory organs of the head. Sight, sound, hearing, taste, and smell are typically going to be categorized, and equilibrium as number 6, are typically categorized as our special senses. So they're going to employ complex sense organs associated with your head. When I was an undergraduate, my anatomy prof explained special senses to me as those things that earthworms don't have. Maybe that'll help you. Earthworms can't see, they don't have equilibrium, they don't have a sense of smell, so on and so forth. When we think of vision, this is the specialist of the special senses. We as human beings are going to get most of our information visually. Just think looking around this room right now. You can see lots of people, and you're learning about them when you look at them. You can't hear them, you can't smell them, you're not touching them, you're not equalizing with them or balancing on top of them. When we look at stuff, that's how we get most of the information we have in our bodies. If any of you go into education, you learn that most people are visual learners, which is one of the reasons why I take so much time to put pictures in all the PowerPoint slides, because pictures are worth 1,000 words. Now, when we think of vision, I want you to primarily associate vision with the occipital lobe. The occipital lobe is named for the occipital bone, and is going to have the primary visual cortex here in yellow, and the visual association area next to the primary visual cortex in green. Now, when we think of vision, we process that vision with the primary visual cortex of the occipital lobe. Pragmatically speaking, this means that when people get hit in the back of the head, they oftentimes are going to have problems with their vision because they're getting a blow or contusion to the occipital lobe. Now, with that said, other parts of the brain help us to recognize what we're seeing. So, for instance, facial recognition is primarily going to be localized within the temporal lobe of our cerebrum. When we think of hearing, the lobe of the brain closest to the ears is primarily associated with hearing. So, we're going to have the temporal lobe is going to be the primary lobe of the brain associated with hearing. We also are going to have the insula associated with hearing as well. The insula is the lobe of the brain that's buried, or the lobe of the cerebrum that's buried beneath the temporal and the parietal lobes. It's kind of hard to get to. To expose the insula, you may remember from earlier last week when we were talking about the insula, you need to separate the temporal lobe and have that temporal lobe pulled down, have the parietal lobe pulled up, and then you would see the insula in between if you separate the lateral sulcus. Getting to the insula is kind of hard, though. Now, when we think of hearing, we have different kinds of hearing that we can do. We can hear spoken words. We can recognize those spoken words. We can recognize someone's voice. We can recognize music. There's all different kinds of ways to process hearing. This is one of the reasons why maybe in the past you've had to sing a song. And if you had to learn a song, because you've used a specific part of your brain to process that music, to process that hearing, you're much more likely to remember it. It's easier to commit to long-term memory. On our figure, I just need to make sure to point this out to us. On our figure for the temporal lobe, our temporal lobe is right here. And we're going to use that temporal lobe primarily for recognizing speech. We also are going to have equilibrium, or in other words, our sense of balance. And there's different kinds of equilibrium. We'll talk about that probably Monday of next week. We have dynamic equilibrium, and then we have static equilibrium. To know how your body is moving and how your body is oriented is equilibrium. And we process equilibrium overwhelmingly with the cerebellum. So when we look at our cerebellum is located back here. It's the little cerebrum with little sulci and little gyri. A well-developed cerebrum is associated with a high degree of coordination, a degree of proprioception. Now, when we look at the association cortex for equilibrium, that association cortex is on the roof of the lateral sulcus. So as we look at our lateral sulcus, here's the lateral sulcus, and on the roof of that indentation, we have the association cortex for equilibrium. When we are choosing to consciously move our bodies, that decision to consciously move our bodies is going to activate the association cortex. So consciousness of our body movements and orientation in space is associated with that. When we think of our sense of taste and our sense of smell, that's gustation and olfaction. Our gustatory center is the part of our brain associated with taste. Our olfaction center is the part of the brain associated with smell. When we look at our sense of taste, it's going to be located or centralized on the inferior end of the post-central gyrus. Think back to your lab terms. We have the central sulcus right here, and then with the arrow in dark purple is the post-central gyrus, and then on the inferior end in orange, we have the primary gustatory cortex. So when diagnosing a patient and trying to figure out what part of the brain the patient had a damage or injury to, the best way to figure that out is to figure out what does the patient struggle with. If that patient struggles with coordination, there's a good chance there's been damage to or stroke to the cerebellum. If they lose their sense of olfaction or their sense of taste, that primarily is going to indicate that either there's damage to that cranial nerve or that part of their brain has been damaged. The best way to diagnose what's wrong with their brain is to figure out what function they have lost in that patient. So if we look at somebody and they have lost their sense of taste, that's a red flag that maybe they had a microstroke or a mini stroke within their gustatory cortex. When we think of olfaction or smell, this is going to be associated with cranial nerve number 1. Cranial nerve number 1 consists of the olfactory bulb. And as we look at our olfactory cortex, that's going to be located right down here. It's on the inferior medial margin of our temporal lobe and frontal lobe. So medial surface of the temporal, inferior surface of the frontal lobes. So let's have a review question. Which of the following is correct regarding the occipital lobe? Is the occipital lobe concerned with mood, memory, and emotions? Is it the principal visual center of the brain? Is it the site for receiving and interpreting general senses? Is it concerned with voluntary motor movements? Or is the occipital lobe likely to play a role in understanding spoken language? We're down to about 30 seconds. If you don't remember, it's okay. You can flip back a couple slides. It worked out well that we paired the limbic system with today's lecture. Answers in, we're down to less than 10 seconds. And it looks like, oh, everyone got them in. So when we think of our occipital lobe, overwhelmingly I want you to associate visual processing with our occipital lobes. And it looks like, as a class, it's fantastic. Let's talk senses. As we look at general senses, the senses that we can have on any part of our body, generally speaking. So touch, pressure, stretch, heat, cold, pain. These are the general senses. And as we look at these general senses in our head, we're going to have cranial nerves. Those cranial nerves that you memorized in lab are going to innervate heat, temperature, pressure, sound. All of the general senses for our head are carried to our central nervous system via the cranial nerves. When we look at the rest of our body, those general sensations are going to be carried to the central nervous system via the spinal nerves that we talked about. And of those spinal nerves, there are many spinal nerves. Now, as those general senses enter the central nervous system from the spinal nerve, they'll enter the spinal cord. From the spinal cord, general sensation is going to be sent to the thalamus. And when we look at the thalamus as it's processing that information, it's going to process information from the contralateral side. This is a term that we talked about when we talked about the spinal cord Friday of last week, I believe. So it's, no, excuse me, Monday, I believe, of last week. So it's been a while. To refresh, when we have contralateral, that term means that the signal has crossed from left to right or right to left. It desiccates. So as the thalamus is receiving that input for general sensation, the right part of the thalamus receives information from the left side of the body and vice versa. Now, as the thalamus receives that information, it will relay that general sensation to the post-central gyrus. So let's look at this map for a second here. We'll go back to the previous slide. Here's our central sulcus. Posterior to the central sulcus in dark purple is the post-central gyrus. That post-central gyrus is where we have our primary somatosensory cortex. This is the primary part of the brain to process general sensation. As we look at this central gyrus, it's going to be located on the parietal lobe. It's the first gyri, or most anterior gyri of the parietal lobe. As we look at the association area for general sensation, it's referred to as the somatosensic association area, and it's going to be posterior or caudal to that post-central gyrus. So as we look at this region of the brain that's right here, this region that's directly posterior or caudal of the post-central gyrus is the association area for our general senses. That information is first sent from the thalamus to the post-central gyrus, and then from the post-central gyrus, the association area helps us to interpret it and make sense of it. Um, on this figure right here, we have a sensory homunculus map. I always think these are kind of fun. A homunculus, for those of you who follow medieval history, and when people practiced alchemy, a homunculus was a little human. The goal was to make little artificial human, and those were referred to as homunculi. The meaning of that word has changed over the last 1,000 years, though, and now when we look at a homunculus, we're going to plot sensory functions over the brain. And we find that not all parts of the body are created equal. We have an extremely large proportion or a high density of sensory receptors in our face, in particular our tongue and our mouth. So as we look at the sensory homunculus, our face receives a disproportionately large amount of our sensory processing power in the brain. We have a ton of sensory receptors in our face. I have little kids in my house right now. And one of the ways, the classic kids learn in the world, whatever they grab onto, they jam it into their mouths. And as they jam it into their mouths, they are using all those sensory receptors of their tongue and lips to gain information about that object. Other parts of the body that have higher densities or higher than average densities include the genitalia, the hands, and the feet. Parts of the body that have a disproportionately low density of sensory receptors are those parts of the body that when people hit you. So think of your shoulders, your back, your thighs, your upper arms. We have a lower density of sensory receptors in those parts of our area. So our sensory homunculus is going to de-emphasize the size of those regions of the body. So as we look at our sensory homunculi, it shows visually the different regions of our body plotted out over the primary somatosensory cortex. When we look at this study of where sensation in a certain part of the body is going to be sent to the brain, this is going to be referred to as a somatotopy. Then this is going to be a study where you can stimulate certain regions of the body and then see what part of the brain lights up. Or you can correlate it to, say, use that loss of sensation to a certain part of the body to have a very strongly educated guess to which part of the brain a stroke has occurred. Let's talk motor control. We just talked general senses. Let's talk voluntary motor movements. So when we think of movement or motor control, our primary motor cortex is going to be located in precentral gyrus. Our precentral gyrus is going to be located, to back up a couple slides here, anterior to the central sulcus. The precentral gyrus is right here in lavender. And that is located within the frontal lobe. It's the most anterior portion of the frontal lobe, most anterior of the frontal lobe. And this is where information from motor movements is going to leave the brain. So when we decide to make a movement, a voluntary motor movement, our part of the cerebral cortex that ultimately senses is going to be the cortex. And from there it goes to the thalamus, and from there to down the spinal cord to that skeletal muscle. We also have association cortex. Our association cortex is anterior to the precentral gyrus. And this motor association cortex helps us to plan the movements. So a lot of times coaches would tell athletes that you need to envision yourself winning, plan your moves, plan what you're going to do in your mind. Whether it's you're a gymnast or a wrestler or a basketball player, thinking about the movements that you want to do will actually help you to do the movements. You're using your association cortex to pre-plan out those firing signals that will be sent from the primary motor cortex. As we look at the precentral gyrus, we could have a homunculi of the precentral gyrus as well. It's very analogous to homunculi of the primary somatosensory cortex with a few alterations. For instance, a notable difference is that in the somatosensory homunculi, the genitals function pretty prominently deep within the longitudinal fissure. We don't have a lot of motor control, voluntary motor control in our genitals, so we don't need to dedicate any of our primary motor cortex to moving our genitals voluntarily. But other than that notable difference, also we don't have a lot of voluntary movement of our visceral organs, so we don't have organs of the abdominal pelvic cavity located down here, like we did in the somatosensory cortex. Other than those two notable differences, it more or less is analogous to the sensory cortex. The parts of our body that have small motor units, or motor neurons that control a small number of skeletal muscle fibers, these parts of our body are going to have a disproportionately large amount of real estate on this gyri dedicated to them. So when we think of the tongue, movement of our tongue is the most highly regulated part of our body. This is the part of our body that has the smallest motor units and will have, per surface area, the largest amount of, or the highest density of motor units. Our motor homunculus is distorted for the same reason that the somatosensory cortex was distorted. So when we think of our shoulders, our legs, our upper arms, these parts of our body have very large motor units. We don't have a lot of control or a high degree of control over those parts of our body. Now, as we're looking at our cortex, there's an important cell type. This is the third time you've been exposed to these during class. We had them on a microscope slide in lab. We talked about them in the last presentation. These pyramidal cells, or triangular-shaped neurons, serve to link the cerebral cortex with the thalamus. These pyramidal cells are typically going to be referred to as upper motor neurons. And these upper motor neurons will have their neurosoma within the cerebral cortex and then send their axons all the way down to the brainstem. And as they send their fibers or axons down to the brainstem, they'll merge and form tracts within the spinal cord, spinal tracts. Within our brainstem and spinal cord, these upper motor neurons are going to synapse with lower motor neurons. So within the cerebrum, we have the pyramidal cell. That pyramidal cell is then going to synapse with another motor neuron within the spinal cord, and then that motor neuron is then going to send its axon to the individual muscle that's going to be activated. To help regulate the activity of these pyramidal cells, we have the basal nuclei. Basal nuclei are going to filter out unwanted action potentials. Or in other words, basal nuclei, primarily made up of the lentiform nucleus, are going to filter out twitches and tremors, those unwanted action potentials activating your skeletal muscles. Our cerebellum also is an important part to help filter out the activity of the pyramidal cells. Our cerebellum helps us to be coordinated. Think about those times when you're tripping and your foot sticks out automatically. That's something regulated by our cerebellum. So let's talk in more detail about the basal nuclei, that region of gray matter buried in white matter of the cerebrum. Our basal nuclei helps us with intentional movement, regulating intentional movements. And I need to underline intentional movements. When we think of damage to the basal nuclei, the classic symptom associated with damage to the basal nuclei is going to be tremors only when you're trying to move your body. They're called intention tremors. And the way you diagnose an intention tremors, you just look at the individual, look at the patient. If they're sitting there and relaxing and they're still when they are trying to relax, but then they start to tremble the moment they start to move their arms or move their hands, that's called an intention tremor. They only tremble when they are intentionally moving their bodies. And that is associated with damage to the basal nuclei. Repetitive movements are highly coordinated by our basal nuclei. Our basal nuclei will work in conjunction with the cerebellum to to allow for us to develop what is known as muscle memory, those automatic motor movements that you don't even think about anymore. Things like tying your shoe, reaching for the shifter in your car, things of that nature. If we have abnormal movement of the body, known as dyskinesis, this is going to be caused by lesions to our basal nuclei and or lesions to our cerebellum. If we look at our cerebellum, more than anything else, is going to be associated with motor control. Our cerebellum has multiple functions, but overwhelmingly I want to emphasize motor control for that part of our brain. So as we look at our cerebellum, it's going to help us to process that information from the pyramidal cells of the precentral gyrus. It will help us to maintain muscle tone. It helps us with motor learning. It helps us with voluntary and involuntary motor movements. Our cerebellum is incredibly important for regulating both smooth muscle and skeletal muscle. When an individual has a lesion in their cerebellum, oftentimes that's going to cause them to have an awkward gait. That awkward gait is a condition known as ataxia. Let's talk about language. If you've had me for lab, you've probably heard me say on oral occasion that you need to look at the material, you need to touch the material, you need to think about the material, and you need to say the name of the material. And we're getting at the reason why. Different parts of the brain are going to be activated when you do those different things. And by activating all of those different parts of your brain, you're going to be more likely to have that information stick into your brain. So let's talk about the Wernicke area of the brain. Our Wernicke area is located right here in bright green. The Wernicke area is primarily associated with recognizing the meaning of words. To recognize the meaning of language is associated with the Wernicke area. And then determining the words that you want to say is also associated with the Wernicke area. So individuals that have a silver tongue that just always seem to say the right thing, it's partly because they have a very well-developed Wernicke area within their left cerebral hemisphere. We also have Broca's area. Wernicke's area is associated with language recognition and language formulation. When we look at the Broca's area, that's going to be associated with the movement of the muscles that you use to deliver the language. So when you move your tongue, when you move your mouth, when you move your lips, and then the inhalation and exhalation associated with activating your spinal, or with activating your vocal cords, all of these motor movements need to be coordinated, and they're coordinated in a region of the brain that's in bright pink right here, Broca's area of the brain. So when you think about the terms, when you look at the term, you activate your visual cortex. When you recognize the term, you activate Wernicke's area. When you tell your study buddy about the material, you're activating Broca's area. By using different techniques, you're going to activate different parts of your brain, and you're more likely to remember that information. We also have the affective language area, not shown in this figure. This figure is of the left cerebral hemisphere. Our affective language area is on the right cerebral hemisphere. And our affective language area is one that I wish was more developed in me personally. It's recognizing tone of voice, giving the right emphasis to the words that you say. Individuals that have a stroke or a lesion to the affective language center are very flat, very words at the time, any dynamic to their speech. They also have a hard time recognizing sarcasm in other people. That's the part of the brain that we use for the, to make speech interesting. I remember during my teacher training, it was emphasized to me, it's not what you say, it's how you say it. How you say what you say is associated with the affective language area. This will be our last slide. Aphasia. So as we look at aphasia, aphasia is associated with any kind of a language deficit. And there can be multiple language deficits depending on what part of the brain had the stroke or the lesion or the bleed. So if we have non-fluent aphasia or Broca aphasia, this is somebody who's having difficulty using the motor sensors to formulate the speech, the language that they want to communicate. These individuals are using words that can approximate the correct word, but they have very slow, clumsy, awkward speech. When I talked to my wife about this stuff, she described individuals that have Broca aphasia as people that slur the words together. They have a because they have difficulty regulating those motor units. Individuals with fluent aphasia or Wernicke aphasia just speak gibberish. You can say something to them, they misinterpret the meaning of the words, and then the words they try to communicate to you mean something different. So if they're speaking clearly and clearly speaking gibberish, it's probably Wernicke aphasia. And then finally, there's anomic aphasia. These individuals can understand speech, but they can't read or identify written words. And this will be our stopping point for the day. We'll pick up on Friday with a review question.